Okay. Can people see the full screen of my slides?
Yes.
Yes.
Okay. All right. I wanted to make this really, really quick here because we're starting a new year and just I want to give an update on what we've done for the past 2 years. I can't believe it's been 2 years. So we, as this working group, we have held a variety of these remote meetings, about six for 2 years. And there's been other things that we've covered in some of these meetings, but the topics in the meetings, just like this one today that we have covered, is starting off with definitions of mixotrophy, how we define mixotrophs and mixotrophy. We had 2 sessions related to biogeochemistry, one of them that was focused on modeling, while the other was more about empirical data, talking about omics and that methodology to study mixotrophs, the biogeography of mixotrophs, remote sensing, using that to study mixotrophs, the evolution of mixotrophs, and today we'll be talking about HABs. And so we've had a variety of different people associated with the working group and from outside the working group come in on talk on each of these topics. And there's hopefully more to come this year. So other things that we've discussed about is looking at temperature responses of mixotrophs in connection to climate change or other aspects of climate change, looking at using the methodology of stable isotopes to study mixotrophs and mixotrophy, and then a more vaguely undefined topic, but something related to food webs and trophic transfer, which hasn't been touched on. And I have question marks here because we are still, as always, taking suggestions of what the community would like to hear us talk about. Hopefully that's what keeps people coming back. And the working group itself, we have hosted some in-person, well, some meetings for the people who are more like the official working group members and working on a lot of other things outside of these virtual meetings. But we have had 2 meetings and plan to hold a third this coming summer. So we had our first one October of 2021, which was, well, kind of hybrid in of itself. It was mostly virtual. And then we had one this past summer, which was mostly in person with a little bit of hybrid, and that was held at Hui. And then we'll have one coming up again this June that is mostly in person, a little bit of hybrid, and that will be held at Bigelow. We've also held sessions at scientific conferences that people can present to. So we've had sessions at the 2022 Ocean Sciences, which was virtual, unfortunately, and not in Hawaii. But we still had a pretty lively session with a good number of talks. That was exciting. And then there's people from this working group who are hosting a session at the 2023 ASLO in Spain. And if you, for some reason, are thinking about attending that meeting and have appropriate research abstracts happen to be due on February 23rd, and this is the name of that topic. So here's the session code that you can search and the name of that session if you would like. They're definitely much towards the bottom, like a long list. As you can see, they're number 112, so they're way towards the bottom. And we're also hoping, whether it's associated with this working group or not, there to have like another mixotrophy themed session at the upcoming Ocean Sciences in 2024. We've also been working on some publications. So one of them is essentially our version of a white paper that's outlining research priorities that this group has worked to synthesize, but it's not a true white paper in that we're trying to get it published at Journal of Plankton Research as a Horizons paper. And we've actually gotten revisions back on it, so we're working to address those revisions. So fingers crossed that this publication will be seen in the first half of this year, assuming all goes well. And then we are working on another publication. This one has a first full draft. But the idea behind this is that we take the research priorities we've outlined and tried to go over how exactly people could do research to address these different questions associated with these research priorities. And the hope is that this will get submitted in the first half or maybe close early and second half of this year to frontiers. And that's everything. I'll stop sharing. This kind of what the working group's been up to and where we're going. And so I'll stop sharing. There we go. So yay. No, I don't really have anything else to say. I just Pat, are you doing okay on your computer there? Are you ready to switch over to, or are you still running into issues?
It looks like Pat has disconnected, so she's probably trying to do a hard restart. So maybe is it okay to have the talks reversed?
We can have a talks reverse because it does seem that Lisa Campbell is on.
Yeah.
Only if Lisa Campbell feels ready to go. Because I know you just joined. I don't want to like.
Yeah,
throw you
no,
away from
I ran up the stairs, but I'm breathing normally now. So yeah.
Thank you, Lisa.
So but we didn't, yeah, so I have my presentation open. Whoops. But what do I have to just share it? Or you
Yes.
have to make me.
Yes, I have made you the presenter, so you should be able to share now.
Okay, wait, I just lost you. Okay. Let me just go here.
Well, Lisa's bringing that up. I can give her a short introduction. So Dr. Lisa Campbell, she is at Texas A&M, and she's been doing a lot of research, well, for a while now on HABs in general, but a lot of her HABs are very dinoflagellate focused, and she's also been doing a lot of work using IFCBs or imaging flow cytobots and using trying to come up with early detection of these dinoflagellate blooms that are very common down in the part of Texas that she works in and in the Gulf of Mexico. So
Yeah.
I asked her to come to talk about early detection of HABs and possible consideration of mixotrophs, as a lot of what she works with happens to be mixotrophs.
Okay, and I practiced last week, but now that I'm not seeing my presentation view, so I'm not exactly sure why I'm not seeing that. So let's see.
So I actually had to do mine twice. I had to go to the share again. I actually had to do it twice to bring it up. Oh, something's coming up.
There you go. We can see it now.
You can see, you see the whole thing?
Yes, it's in full, it is in presenter, yeah, presenter view, full screen.
Okay, and then I'm in my notes here, so, okay. So I've got to go back then to you see my opening slide now? All right.
We do.
So,
take it away.
All right, early warning. So take it away. All right, in this talk, I'll first describe the HABs that are concerned in Texas and what the requirements are for successful early warning. And then I'll introduce the imaging flow cytobot, although the details of this are beyond the scope of the 15 minutes. So but I'm happy to take questions afterwards. And I'll show that the continuous operation of the automated imaging flow cytobot has been successfully deployed on the coast of Texas. And then what we've learned about dinophysis, and I'll show you some examples of the time series, and then work that we've been doing to identify factors that limit the bloom initiation and model development for determining bloom initiation. And this is work that's in progress. And so what our future goals are, and then of course take input from all of you. Okay. Next slide. Let's see. All right. So in Texas, we have Carenia brevis, which is shown here in the left image here, which of course is the Texas red tide, which is the one that has caused a lot of issues over the last many decades, and it causes dead fish to pile up on the beach. And then of course for the humans, the main route of exposure is through ingesting shellfish, and in Texas, of course, oysters is the main route of exposure. But of course, more recently, we've also had Dinophysis ovum as a important HAB species, and brevis pretty much is a fall bloomer, and whereas Dinophysis typically Valentine's Day. So tomorrow is the day when if we see it in the water, we're going to have a bloom. And so we're waiting to see if we'll have a bloom this year. But we found that early warning is the most effective mitigation to predict harmful algal blooms. And usually, I mean, I don't need to probably tell everyone what a HAB is, but usually an unusually high concentration or of toxic or nuisance algae. And really the requirements for a successful early warning system are regular sampling, high frequency sampling, and frequency with high accuracy of detection. So the imaging flow cytobot then, which was designed by Heidi Sosik and Rob Olson at Woods Hole, is then answers all of these objectives because it can be operated continuously and autonomously, and it takes a 5 ml sample every 20 minutes, so we get about 70 samples a day, and then using this instrument, which combines flow cytometry and video technology, we image all the plankton in the water, and then using facial pattern recognition software, which software that's similar to facial pattern recognition, we can identify the particular types. And I'll, again, not have time to go into all the details of this, but I just wanted to emphasize that this is unattended and continuous operation. And so where we have our networks here set up, we have a station at Surfside Beach, which is just due south of College Station, really. Where I am is just a little bit off this map. And then also down at Corpus Christi Bay here, the Port Aransas facility up here. This channel into Corpus Christi Bay has been a very important sentinel site because at high tide, the whole Gulf of Mexico enters this channel. So it's been very successful observatory point. So the acronym for our network is TOAST, Texas Observatory for Algal Succession Time Series. And so this has been operating in Texas at Port Aransas since 2007, and then the Surfside Beach Station has been operating since 2017. So in this more than 15-year time series, of course, we have maybe 2 billion images or more. So obviously automated classification is essential. And so this is where the automation and classification has really, really been important. And so here are some examples of imaging. Here's Karenia here and Dinophysis down here, and then a lot of the other groups that we've developed classifiers for. And the approach, this Heidi developed a random forest approach, which is a Bayesian program that we've then modified a little bit, and we're now using a convolutional neural network, which has improved, which is what I was alluding to earlier, which is similar to the facial pattern recognition software that Google uses, for example. And with this approach, we've been able to distinguish Dinophysis a lot more easily. And here's Perocentrum texanum, which is blooms concurrently with Dinophysis, and with using this new approach, which Darren Henricks published in 2021, we now can distinguish as we have less confusion between these two species. So that's an advantage of using this approach. So I just wanted to show you then the automated classification and messaging system. So we download the data from the field to my lab here in College Station every hour, and then we run the image processing, and this is all automated. And when we detect cells of either harmful algal, either Karenia or Dinophysis, when the cells are greater than 2 cells per mil, we send notifications both to my lab and to local state managers and the Texas Department of Health Services, which is the agency responsible for closing the oyster fisheries. And this is just a sample of an email that gets sent out. And this was for a Karenia bloom, but we have the cell counts for the last 10 hours and then a little plot. So that, and then with links to our website. This is, again, this was a Corynea bloom, not Dinophysis, but you can see then that when you go to the website, you can look at the cells that are blooming that past hour, and then you can see that, yes, in fact, that is Corynea. And of course, all the state agencies are able to recognize both Dinophysis and Corynea very easily. So this has been then in operation. And with this system, we've had 8 successful early warnings, and no illnesses have been reported due to harmful algal bloom. So we feel this is very successful. So looking at Dinophysis, what have we learned? So this was within the first 6 months of our initiation of this early warning system. We did have a Dinophysis bloom, which was the first bloom that was reported in the United States. And the red dots here are the weekly sampling by Texas Parks and Wildlife. And then this is then our continuous daily averages of cell. The black lines are the. Can you see my mouse?
Yes.
Oh, okay. So you can see we did have a gap, which happens periodically due to some issues, but we see we could first detect it here, and but sometimes, of course, due to the patchiness of the populations, sometimes this field sampling, of course, do not detect it, whereas then the continuous operation we were able to detect Dinophysis, and then but the advantage, of course, for having this early warning is that you can give the Department of Health, the managers, the state managers who have to go and sample early warning, so that they can set up all their assays and their field boats and personnel, and with several weeks notice, advance warning before the cell counts get to the bloom conditions where we really start worrying about human health. So, and again, this makes the whole sampling procedure more efficient because they know when and where the blooms are occurring. So this was when we first reported it, and then they sampled it and detected it in oysters, and then they were able to close the oyster fishery, which was an advantage because the following week, there was an oyster festival, which occurs every year on the coast in Texas, and they have oyster eating contests. So if 20,000 people attended this oyster eating contest, then of course a lot of people would have been sick. So this was averted a potential disaster. And then another advantage of having this time series is, of course, then we can tell when the bloom is over. Towards the end of April, then it's detected at much lower levels. And so then again, this helps the Department of Health with their sampling. So what we know, of course, about Dinophysis is that it's mixotrophic. Dinophysis produces the okadaic acid, but it consumes mesodinium, and then which in turn has consumed cryptophytes. I won't go into details because I assume that this audience is very familiar with this mini little food web. But when this is our time series from this imaging flow cytobot at Port Aransas, and you can say this was the first bloom. And of course, so then because we saw this bloom, we were all ready the next year. And of course, HAB scientists always, when you're ready to look for a bloom, they don't occur. So in 2009, we didn't see one, but in subsequent years, we have seen a number of blooms. Some years we don't, some years we do. Oops. So this, we wanted to know, though, if we could use the prey item, the mesodinium, as an early warning symbol for Dinophysis. And so in 2008, as you can see, blue, this is the time series of Dinophysis, and red is mesodinium. And mesodinium had very high numbers about a month ahead of the Dinophysis peak. So that was very enticing to us to say, can interactions be a predictor for Dinophysis? And so this is 2008, but we know that in 2010, the blooms occurred later, and then every year there are different peaks or different timings of the phenology of the blooms of Dinophysis ovum at Port Aransas. So what we decided to do, though, is to look at the physiology of the different groups. And so we did a matrix of temperatures which is shown on the x-axis, and then each column is a different salinity for each of the prey items and dinophysis. But don't have time to go into all the results, but I just wanted to look. We looked at the growth rate at each combination of temperature and salinities, and then use these parameters in a model for early warning. And so I just want to highlight that the results for dinophysis, and the open symbols are dinophysis ovum, which occurs here in the Texas, the coast in the Gulf of Mexico. And acuminata, we used Juliet Smith's isolate from Virginia, are the filled symbols. So, but just looking at the open symbols, you can see that the temperature range basically was between about 18 and 25, and any temperatures above 25 for both species, there was no growth. So we then looked at the last 10 years of data, and in years when there was a bloom, we looked at temperatures, and then in years where there was not a bloom, and the temperature increase, it appeared to correlate well with when the timing of that phenology that I showed in the previous slide occurred. So basically in the model that we in a cold year where the water didn't warm up until later, then the bloom was later, say in 2010, whereas in 2008, when the water warmed up earlier in the year, the bloom occurred earlier. So we think temperature is a very important driving factor for when the dinophysis do bloom. And so we hope, this is just a goal, but we hope we can put the factors, temperature and salinity, into when teleolycs will grow, and then mesodinium, and then dinophysis, and the combination of these factors, and then combined with coastal circulation models, like such as the HICOM model in the Gulf of Mexico, and then predict perhaps where dinophysis will bloom, when and where it blooms. And this is just a work in progress by my former grad student, Jim Ferrandino. But of course, there can be other factors than temperature and salinity. Can be food availability, prey specificities, prey size, which is a work that Jim is working on currently, the dilution rates and rescue rates, so when new cells are added to the population. And so this is just an example. In 2008, we had a number of, in terms of competition, a number of other Porocentrum species. And in 2010, and then in 2011, we only saw dinophysis. So we don't know if competition also may play a role. So I hope to get some feedback from everyone. And I just want to show that how valuable the imaging flow cytobot has been for the study of mixotrophic dinoflagellates. You can see that here's various examples of engulfment. I love this image of this gyrodinium having ingested a large diatom. Then we also examples of pallium feeding and peduncle feeding. And the advantage, of course, with the imaging flow cytobot is that you see live cells in action and not the artifacts due to fixation that may plague studies of mixotrophy. So with that, I'd like to conclude. So I've showed you that we've established a HAB network, TOAST, and we can use a CNN, a convolutional neural network classifier, for improved identification, especially of HAB images. And then we have an automated alert system that provides early warning. And then we can use the predator-prey interactions and the lab experiments to determine growth rates that have fed into model parameters. And we hope that this can be used in combination with circulation models to predict when and where dinophysis may originate. And then future goals would be then improved prediction of harmful algal blooms with community dynamics. And then again, we're working with a group on an EcoHab project to study the metatranscriptomes queued, perhaps by the time series. So with that, I'll take any questions. Thank you very much for your attention. I just want to acknowledge all my colleagues in the development of this, Darren Henricks here and at Texas A&M, and Jim Ferrandino, my former student, and then funding agencies. Thank you. And yeah, I guess then we were asked for questions. I don't know the protocol here, but I can take questions or not.
there we go. Sorry. Yes. Yes, we do have time for a couple questions. If anyone would have anything for Lisa after that very, very interesting talk.
Should I minimize this or stop the program?
You can, if you want to stop share. I guess I'll start things off with a question. Um, have has your lab or group tried to do any detections of uh, teleolex or mesodynium in relationship to early detection of dinophysis, or is that, do you think, a possibility?
Yes, we have classifiers for both those groups, and I had a student a number of years ago looking at relationships and using time lags, but there was just a weak correlation if you used based on temperature. So we think that's why we went to look for other factors that may control the timing of the blooms. But yes, we can plot all three populations, and there's no strong, I mean, when there's a large number of mesodynium, isn't always a large dinophysis bloom, and if there's a large dinophysis bloom, we don't always see mesodynium preceding it. That's sort of what your question was asking.
Yes, yes, thank you. I'll take that
It's
two. Oh, go on.
yeah.
I was thinking we've got two questions. George, I think I saw your hand up first, maybe.
Yeah, thanks. Thanks, Lisa. Great talk. I'm really interested in the project. I wondered if the dinoflagellate changes in its toxicity over time as well. Do we know anything about that? And is there monitoring for that?
Yes, that we do monitor. That's sort of one of the goals of our current EcoHab project. We've been doing for the last 4 years. We've been sampling the cells over time, and I mean over the time of the bloom. I don't know that we have any answers to the question yet, though. I don't think there's nutrient results impacts on the toxicity, but for now it's mostly species dependent differences. So for example, Dinophysis ovum is only produces ocadaic acid, and it produces about 10 to 100 times more than Acuminata. And so it's more species. So in for example, on the West Coast in Washington, there's a mixture of different species. So it will depend, the toxicity of the bloom would depend more on the composition of the population rather than nutrients or any other, or time series of the, of the longevity or whatever of the bloom. I think that now that we may find more things as we, look into our time and do more of our analyses of our time series. But right now, I think it's more of the species differences. Does that answer your question?
Yes, thanks.
I was going on the line here. There we go. Looks like Susanna Leles has a question.
Okay.
Sorry, Susanna,
Yeah.
I heard your hand went up before George's. It's all very weird on here.
Thank you, Nicole. Hi, Lisa. Beautiful talk. I really enjoy it. And I love your images, and it's very interesting that you can actually see the trophic interactions happening, right? These dinoflagellates ingesting different prey. And I was wondering, do you think if we could use your images, let's say if you have enough number of mixotrophs that you captured with the flow cytobot across different seasons, for example, and then if you focus on a certain species, let's say they're in summer, you can see a lot of cells that ingested prey, and then maybe in spring, you don't see a lot of that. So do you think you could use these images as a proxy for how much ingestion is going on for these mixotrophs?
Yeah, that's a very good question. And you have to remember that it's a computer looking at it, not your eye. So what I would say is that, yes, we can pull out, say, all the dinophysis, but to determine whether they've ingested things like I showed in my slides versus non-ingested, that I think you'd have to do manually. But certainly if you know, if we pulled out all the Dynaphysis images, then then you could do that manually in us. But I don't know that you could train the computer to, well, I don't know. You just have to try, perhaps, because you need about 500 to 1,000 images to effectively train the instrument to distinguish the different groups. So versus say an empty Dynaphysis versus a one that had eaten a lot. I would say as a first cut to answer your question, I would do it manually and then try to do it automatically. But I think that's a really important question. I think that would be really fun. I could send you a time series of all the data if you want to look and see. Yeah, that would be great.
That's amazing. Thank you so much.
All right, and we've got one more question. Isad, if you would like to ask your question.
Okay.
Oh, sorry, you're still muted.
Barry. Still muted.
Oh no, it's still
maybe put the question in the chat.
Heather, are you unable, are you able to unmute him?
He says that it says that he is unmuted, but he might be tied to a headset instead of a computer.
Yeah, right. Yeah.
Speaker
Maybe he
system,
can just put his
so
question in the chat. But while he's figuring out, I wanted to know why do you think Mesodinium are such picky eaters? Apparently they'll only eat these teleolocs, and so
Oh, that will be for our breakout rooms. So we breakout rooms after this. Yes.
Oh,
yes.
okay. I'm
And
sorry.
people
I
will
missed the
discuss that.
beginning. I missed the beginning introduction, so I don't know. Okay, sorry. So I'll close this.
Yes. All right. So yeah, Isa type his stuff into the chat and Lisa can address it in there. But we can move on to Pat, who thankfully has fixed all of her technical issues. So we will actually be able to hear from her, or hopefully most of her technical issues. Yeah, Pat, if you want to, are you able to share your screen now and get that set up? And while you're getting that set up, Pat is a professor at the University of Maryland's Center for Environmental Sciences, or Environmental Sciences Horn Point Laboratory. And she has done a lot of work with phytoplankton and nitrogen. But she has also been doing a very, very recent project with some collaborators looking at modeling a mixotroph in Chesapeake Bay. And I think it's very interesting work, and I'm really happy for her to talk about it here.
Thank you, Nicole, and apologies for the snafu. Timing is everything, right? Crashes occur. Again, my apologies. So I do want to talk today about our mixotrophic model of Carlidinium veneficum in Chesapeake Bay in the belly of the beast. This is most definitely a collaborative effort with Ming Li from Horn Point. Most of you know I am not the modeler, but it has been a real pleasure working with him on this project. As most of you know, Carlidinium is a common HAB dinoflagellate in the US and globally. It's an estuarine species favoring mid to low salinities. And you can see in the Chesapeake Bay, it blooms typically mid-bay and upper bay. And it also prefers warm temperatures. So we see it in June and July. During the warmest month of August, it tends to be a little too hot for Carlidinium, but we may see some blooms again in the fall. And it is a constitutive mixotroph. So we've been looking at modeling mixotrophy in Carlidinium using the model that was originally developed by Kevin Flynn and Aditi Mitra, the perfect beast. The perfect beast model has 8 state variables. It describes carbon, nitrogen, and phosphorus, as well as chlorophyll within the mixotroph itself, as well as in the food vacuole after the mixotroph has fed on algal prey. So our first exercise with this model was to model some laboratory experiments in which we varied the nitrogen to phosphorus ratio of both the predator and the prey. And you can see here the model was able to fit experimental data quite well. And that laboratory data also gave us insight into how biomass changes under different NDP conditions. Shown here are the high NDP treatments, and you can see for temperatures, the warm temperatures, 20 and 25 degrees, Carlidinium as a mixotroph, shown on the left, reaches higher carbon biomass than when grown as an autotroph. So these results gave us courage to try to develop a 3D model of mixotrophy of Carlidinium. So the mixotrophy model is embedded in a biogeochemical model that I'll say more about in just a minute, and that's embedded in a ROMS model of Chesapeake Bay. So the biogeochemical model was originally developed for exploring hypoxia in Chesapeake Bay, and it includes compartments such as phytoplankton, nitrogen, phosphorus, silica, DOC, and POC. It's based on 2 different phytoplankton groups, a winter diatom group and a summer general nanoflagellate group. It also has a 2-layer sediment diagenesis model. And as I said, this has been well used in explaining and predicting hypoxia in Chesapeake Bay. So in coupling the perfect beast model to the biogeochemical model, an additional phytoplankton group is modeled, and that is Carlidinium. So Carlidinium is modeled as a species against the backdrop of the 2 general functional groups of phytoplankton. And this process again is repeated with the cryptophyte prey. So let's jump into some of the results. Here is the predicted versus observed Carlidinium distributions. And what you can see is that it reproduces Carlidinium distribution quite well. It increases in June and July, begins to wane in August. The dots are data from the Chesapeake Bay program. Here's the along channel distribution. And again, you can see by month, highest concentrations developing in the upper bay in June. Then by July, well established and throughout the water column of the upper bay. And again, begins to fall off in August and September. Here's the predicted versus observed distribution of cryptophyte. And the RCA model does quite well also with cryptophytes. You can see their biomass increasing in June, July, and staying up through the fall months. Also shown here in the black and white are data from an earlier study from Aisha Lee and Diane Stecker, showing distributions in July and September of cryptophytes. So here's the mixotrophic feeding relationships And this is where the perfect beast model allows us to look at ingestion, digestion, and the food vacuole content. The first column are the ingestion rates, the middle column are the digestion rates, and then the first couple of panels, May and June, ingestion rate is high, especially in the upper bay, and ingestion rate is higher than the digestion rate. And that leads to a enlarged food vacuole content. Later in the summer, the bottom panels of August and September, here you can see ingestion still continues, but the digestion rate is higher just simply due to increased temperatures. So the food vacuole content shrinks. Again, shown in black and white here are some observed ingestion prey per carlidinium, again, based on Aisha Lee's earlier studies. The model also allows us to look at the distribution of nitrogen and phosphorus. And here we begin to see some interesting patterns. And that is that seasonally, inorganic phosphorus becomes depleted before inorganic nitrogen. So we begin to see inorganic phosphorus begin to develop in April, May, and June, whereas we don't see nitrogen becoming depleted until later in the summer. And the time series on the top plots that as well as river discharge. So carlidinium blooms begin to occur during this period when inorganic nutrient begins to run out in the estuary. So we can look at these factors affecting ingestion rate, and again, in comparison with some of those earlier studies of Aisha Lee. The model results in the top, shown for 2 of the Chesapeake Bay stations, tell us that ingestion increases with prey abundance, which makes sense. And ingestion rate drops off as the P to N ratio increases. The lab results on the bottom, again, are those from the earlier studies of Aisha Lee. So the model is telling us very much the same relationships as we've seen in the lab. We can look at digestion rate, and the model results tell us that digestion is a hyperbolic function of the food vacuole content. And that was seen also in the laboratory experiments. And also that digestion rate increases as a function of temperature. So we can put all of this together and look at the time series of various factors contributing to the transition of autotrophic growth rate to heterotrophic growth rate. Starting at the bottom panel, again, you see that shift in depletion of phosphorus, which occurs earlier than that of nitrogen in the bay. Phosphorus becoming depleted April, May, and June. And it's those months that the cryptophyte prey begin to increase, as shown in the middle panel. And so in the upper panel, you can see that the carlidinium, although its abundance is very low early in the year, it's really growing autotrophically. And then in the middle of the summer, it shifts to becoming a mixotroph as nutrients become depleted and prey become available. So we've used these models to ask a couple of other questions. What happens if we reduce the nutrient footprint, or how will carlidinium change with changing climate? There are a number of climate models that have been applied and downscaled for Chesapeake Bay, and I'm not going to go into these, but just to show that a number of different models have been used. And shown here is the results of one of these scenarios, and that is that as waters warm, the good news is that carlidinium becomes less abundant in the summer, and the peak of the bloom is shifted seasonally. It shifted earlier in the year. Cryptophytes are less affected by these temperature changes. So that's the good news. On the other hand, if we look at effects of nutrient management, using the 40% nutrient reduction target of the Chesapeake Bay program, we can see that there's very little effect on either carlidinium or the cryptophytes. Cryptophytes are reduced a little bit, but the nutrient reduction has little effect on carlidinium blooms because of their alternate nutrient acquisition strategy. So the ROMS RCA mixotroph model captures the observed seasonal timing and the spatial distribution of carlidinium blooms, and reproduces the laboratory findings with regard to factors controlling ingestion and digestion. Mixotroph feeding appears to be triggered by phosphorus deficiency in the bay, and autotrophic growth dominates in spring, but heterotrophic mixotrophic growth dominates in summer. And warming due to climate change reduces the bloom size and shifts it earlier. On the other hand, nutrient management appears to have only a moderate impact on the bloom size, as mixotrophy provides a buffer to nutrient management. So I want to thank my lab group and Ming Li and his lab group. The results here were published recently in this paper shown at the bottom here. And if I can take one more second for a shameless plug, I want to introduce to you, if you are teaching phytoplankton ecology, this book will be coming out later this year. So that's it.
All right, thank you, Pat. Great stuff. Of course, very interesting. We'll have time for a couple questions before we go into breakout groups. So maybe one or two quick questions. It's actually really hard. I think I saw Susanna's pop up real fast. She's got a great question. She's got a question ready to go.
Yeah, thank you, Pat. Great talk and massive modeling work. My question is actually quick. So about the modeling part in the warming scenario, you mentioned they become less abundant, carlidinium. I was wondering why. Is it like because of top-down control, and also how warming would affect the metabolism of the mixotroph in the model?
Good question. So the temperature of the bay actually exceeds the temperature optimum of growth for K. veneficum. It is a direct temperature effect based on the model. And the other question was the temperature effect on digestion. What was What was the second question?
Yeah, no, thank you. That clarifies a lot. The second question was more about, does temperature affect photosynthesis and ingestion rates differently?
So the model has a temperature of, there is a temperature relationship with regard to ingestion and digestion. But these results are output of the model.
Yep. No, thank you, Pat. Yeah, that's exciting.
I am muted. Great. Are there any more questions? It's really hard for me, sorry, the way it's set up to see hands raised. All right. Let me get that set up. So I think we'll move into breakout groups, because now is the time on the schedule to move into breakout groups. And I am putting into the chat to share with everyone our discussion questions for this time. We will have 3 breakout groups, I think, this time. And we've got 4 discussion questions, and we will stay in the breakout groups until 12:15, and May will close them for us. So just about 20 minutes or so, May, and then we'll come back and we will have a report out from each group. All right, there we go. The breakout groups have started. The computer just told me I was muted. So welcome back, everyone. And I'd like to thank again Lisa and Pat, both for their talks, but also for their questions. I thought they were great questions with a lot of great discussion. And I just want to start with breakout room 1, which looked like it had Sarah and Lisa in it. And so if one of you guys would like to talk about what you talked about with those questions, anything you want to mention. Oh, Lisa, I think you're muted too.
I said, I'm going to let Sarah talk since I've already talked.
Okay, so she's doing that because we went off script completely. But it was still a very fruitful conversation. So Lisa and I come from opposite ends of the mixotrophy spectrum, right? So I'm a versus, these constitutive versus non-constitutive forms. And so we discussed a lot about how the idea of picky eating really goes across the spectrum, even the bacterivorous and algarivorous forms. And it really gets to the heart of how these things can accommodate for picky eating, and really looking at their balances between autotrophy and heterotrophy. I think that was the bulk of our conversation. We did talk a little bit about the diatom dinoflagellate ratio as indicators of ecosystem health, and a little bit about trophic upgrading and how that dinoflagellates might actually be good food for higher trophic levels because they integrate mixotrophs. Yeah, keep smiling, Nicole. I don't know, Lisa, did I miss anything? Okay. She's muted, but she's giving me a rave review.
Yes, I'm giving you a rave review. No, I really was interested in the fact that you found that the heterotrophic nanoflagellates are also picky eaters.
All right, thank you guys. Thank you for mentioning too about the diatoms and dinoflagellates as water quality indicators. That wasn't something that we discussed, but of course that comes up a lot. The other group had everyone, almost everyone in it. So do you guys have someone you picked to report out?
No, we didn't pick, but I'll just maybe start reading from my notes, and then yeah, we had George, Pat, Susanna, myself, and Esat. And so any of the other folks within this group, if there's something you want to add, please jump in. So we started talking about prey selectivity, about how dinoflagellates are able to eat prey, you know, as big or bigger than them, and kind of talking a little bit about the mysteries of why they select, you know, why Mixotrophs select certain types of prey. What is it about their the prey themselves that are beneficial? For example, George has this model ciliate that consumes both chloroplasts and, or sorry, chlorophytes and cryptophytes, but grows more with one than the other. So why would it eat cryptophytes at all? And what's the relative balance as we, you know, think about doing more experiments between prey quality versus quantity. In terms of modeling, maybe easier to model prey quantity rather than qualities, but as Lisa noted, you can use proxies of different nutrients for quality. That may be really useful. We talked about cells with different pigments that are sought out for different reasons. Phycobilin for nitrogen, for example. I missed some aspects of this conversation, so feel free to jump in, folks. We also talked a little bit about, you know, how to extend these models out further. Ming-Li is working on a model of Carina brevis as well. You know, that's present out on the shelf that consumes primarily Synechococcus. I noted that, you know, for heterotrophs, for zooplankton, the difference in prey size would result in differences in resiliency under climate change scenarios. And you do see in the empirical data for Crinia, as Pat noted, that there's larger cells in the winter versus summer, as summer cells just appear to be more stressed. And then we had some questions from Susanna about lipids, but we didn't fully finish that whole discussion. Did I miss anything?
No, that was great. Yeah, we spent a few minutes talking about one reason why picky eaters may be picky eaters is digestibility of different types of pigments. So spent some time talking about pigment proteins versus lipids and what that means. So phycobilins, for example.
All right, great. Thank you guys. And so the third breakout group was myself and Natalie, and I volunteered Natalie to do the report out for us. So I'm going to turn it over to her.
Yeah, so we started off with that second question about the diatom to dinoflagellate ratio. And we noted that dinoflagellates, if they dominate, it could be suggestive of mixotrophy, but we really don't know the degree that they're engaging in phototrophy versus phagotrophy. But it could be a starting point and the focus of future research. We also noted that dinoflagellates tend to not be mainly phototrophic based on some of the characterized ones that have been in culture, like chlorophytes, which we know they can lean towards mainly phototrophic. And we moved to question 3. We took a look at if dinoflagellates are consuming large prey, what is the physiological impact of that? And so we noted that to ingest large prey, that takes up a lot of space intracellularly. Some of the photos that Lisa showed, I think, demonstrated that well. And so we were just wondering what the structural investment of that is. Do they have the same kind of cytoskeletal proteins in use to expand and contract like that? And is there an energetic cost of those larger food vacuoles as opposed to the ones that can accommodate small prey? Or are they the same thing? We weren't sure. And then how does that energy investment compare to the use of a peduncle? And then we also talked a little bit about prey selectivity and size. So if a mixotroph can graze on bacteria, how can we generalize that to a lot of the bacterial community that might be present in a certain ecosystem? And if they prefer bigger prey like Mesodinium does that mean that generally they tend to be more specific in their prey choice? And then how specific does it get? Can predators tell apart prey at the genus level, or is it broader than that? And then for that last question, we talked about why dinoflagellates that seem so similar are different in their food choices. And we think that they likely have diverged over evolutionary time, and you might be able to see that on a genetic level. And we note that within a genus, some species show mixotrophy and some don't. Biogeography likely plays a role. And then we also were commenting on the lab studies that have been done with mixotrophs that have shown that they can lose the ability to be mixotrophic after a number of generations. So yeah, lots of questions asked in response to these questions.
Yeah, yeah. I just wanted to add on to one thing that Natalie said, talking about the peduncle or the feeding veil, and with the larger prey, that's some like things feeding on larger prey. That's something that doesn't come up too much. I think a lot of research when you're talking about mixotrophy is just talking about the engulfment of it, but that use of the peduncle and the feeding veils aren't things that come up a lot in conversation. And there's definitely stuff to consider there with like how that saves space in the cell, but then at the same time, you probably have to maintain that feeding veil or peduncle, or put energy into getting it out of the cell and things like that. But it's an interesting thing. But of course, all interesting. And I think what was really great was how everyone had slightly different discussions based on each of the questions, which is extra fascinating. Everyone came in from different points of view and had different topics, because they were questions that allowed for something like that. I hate this part because I'm really terrible at wrapping up meetings. Thank you to everyone who stuck around and partook in the discussion. Once again, thank you for the wonderful talks that we had. I think that was a great way to kick off our 3rd year of doing this. And we will be in touch with what we decide our topic for it should be May is going to be, once we have done that and found speakers to line up with that. And yeah, thank you.